The pair rule genes set up the framework for the next set of segmentation genes, which are the segment polarity genes. The maternal effect, pair rule, and gap genes are only expressed temporarily in the embryo. So the role of the segmentation genes is to define the boundaries of the pair segments, which are also the boundaries that determine different cell fates. The expression of the segment polarity genes begins right after nuclei become separated from each other by cell membranes. Because cellularization has already occurred, the segmentation genes have to code for different types of gene products, including paracrine factors that will be able to diffuse to nearby cells. Prior to that stage, nuclei are still sharing the same cytoplasm, so transcription factors are able to act as morphogens. As the embryo progresses through the pair rule stage, cell membranes separate nuclei from each other, so the embryo becomes a cellular blastoderm. So this means that the segmentation genes are going to be acting in a cellular rather than a syncytial environment. And that explains why these genes have to include signaling proteins, but also receptors, intracellular signaling proteins, and transcription factors. The presence of paracrine factors is what's going to enable communication between cells now that membranes have formed. The segment polarity genes are expressed in a pattern of 14 stripes along the anterior-posterior axis of the animal. Remember that the portion of the fly that's going to give rise to the most posterior region is actually located under the head for a period of time during gastrulation. The segmentation genes are going to be important for establishing cell identity within parasegments. The segment polarity genes belong to the Wnt and hedgehog pathways, and what controls their expression in part is the expression of the pair rule genes, as we'll see in a moment. The Wnt and hedgehog pathway genes will be expressed in an alternating pattern along the anterior-posterior axis of the embryo, so that a row of cells will express hedgehog and the transcription factor engrailed, while the row of cells next to that will express wingless, which is the homologue for Wnt and Drosophila. This particular expression pattern is controlled by the expression of the pair rule genes. So the transcription factor engrailed, which is shown here in the purple, is expressed in rows of cells that either express the pair rule gene FTC, which is shown in blue, or the pair rule gene even skip, which is shown in pink. Wherever the concentration of either of these transcription factors is highest, engrailed will also be expressed. So because these pair rule genes are expressed in 7 alternating stripes along the anterior-posterior axis, then engrail will be expressed in a total of 14 stripes. There are other pair rule genes which repress the expression of engrail, and that explains why it is not found in every cell of the embryo. So engrail becomes expressed in the anterior compartment of a parasegment, which corresponds to the posterior compartment of a segment, while wingless is activated where there is little or no FTC or even skip, but where you have a different pair rule gene. So the pair rule genes set up a pattern of expression of segment polarity genes where at this particular stage of development, the row of cells that constitutes the anterior compartment of a parasegment, which is also the posterior compartment of a segment, will express engrailed and hedgehog, while the cells anterior to the engrail and hedgehog expressing cell express Wnt. Once the pattern of expression of the segment polarity genes is created by the pair rule genes, it has to be maintained throughout the lifetime of the organism, and that occurs through reciprocal interactions between the cells that express Wnt and hedgehog. The transcription factor engrailed induces the expression of hedgehog. Hedgehog is then secreted, and it binds to its receptor in the cell that's immediately anterior to it, so in this cell. That drives the expression of wingless, which is secreted from that cell, and then it binds to its receptor on the cell that is secreting hedgehog. The activation of the Wnt pathway in the cell that is secreting hedgehog actually leads to the expression of engrailed and hedgehog, so this helps to maintain the expression of these genes. So this basically acts like a positive feedback loop for maintaining the expression of these segment polarity genes throughout the lifetime of the organism. Since Wnt and Hedgehog are both paracrine factors, they form gradients as illustrated here. And cells will acquire different fates based on the concentrations of Wnt or Hedgehog that they receive. This can be seen in the epidermal structures that form as a result of the exposure of these cells to different concentrations of Wnt and Hedgehog.